Okay, this next lecture is going to be on cholesterol. And so cholesterol is a molecule that your body can make. Your body has a hard time dealing with cholesterol after it's made. And if you take in a lot of cholesterol in your diet, you're going to reduce the production of cholesterol. So cholesterol negatively regulates cholesterol synthesis. So we're going to learn how cholesterol is synthesized, and then we're going to talk about how you generate steroid hormones from cholesterol. We have the cortisol, we have your testosterone, estrogen, etc. that can all be made from cholesterol. So for this first part, we're going to talk about the steps of cholesterol synthesis. Always occurs in the cytosol. We're going to talk about what is actual free cholesterol and the importance of cholesterol esters, as well as how they are transported, what type of particles pick them up, how do the particles deal with them, and how do they return them back to the liver. And then, of course, we have to know how cholesterol is removed from cells, and then what happens when the regulation goes wrong and you start to get buildup. All right, so just as an overview, of course, cholesterol is the most publicized lipid due to its strong correlation between its incidence in the blood and the incidence of cardiovascular disease. So cholesterol, as you know, is important in our bodies, and you need it in your cell membranes, for example. It also makes your steroid hormones. It also is the precursors for bile salts, bile acids, and it is not required in our diet. Most cells can synthesize this. Okay, so cholesterol is going to be a 27 carbon compound. And all carbons are provided by a single precursor, which is acetate. You are basically going to generate these isoprene units, which are the intermediates. And isoprene units can also be used to make other lipids. They don't have to be used to make cholesterol. So it's not, if you make isoprene, doesn't mean you have to make cholesterol. And isoprene polymerization is going to be the same regardless of what pathway goes down, cholesterol or other lipids. So we're going to talk about the synthesis, transport, uptake, regulation, as well as some of the defects. So the first thing is the synthesis. So you can see here that we have this 4-ring compound, and this is the parahydrocyclopentaphenanthrene nucleus. So this is basically the nuclear core of cholesterol. And you can see the numbering system. So you have the number 1 carbon on the A, and then it's going to go around here to the number 5. And then the B will finish up at 10, and then the C will go through 14, and then 15, 16, 17, then you close the loop at D. Now the structure is going to contain 4 fused rings. So it's an alicyclic compound. Now, free form of cholesterol is where you're going to have 27 carbons. And at carbon 3, you have a hydroxyl group. Just shown here, our 3-hydroxyl group. Between carbon 6 and 5, you have a double bond. At carbon 17 is where you have a member hydrocarbon chain attached. Remember, carbon 17 is still part of that nucleus. And at carbons 10 and 13, you have methyl groups. So here's our methyl group at carbon 10, and then we have another methyl group at carbon 13. Okay, so this is cholesterol. And all of these carbons are going to come from acetate. Now, cholesterol is going to be found in the plasma, and it's going to be found primarily at what we call a cholesterol ester. So 2/3 are cholesterol esters. Okay, and you can see that that OH group at that position 3 was replaced with this ester group. And the R, of course, indicates however many carbons. Now, this ester is usually going to be made up of linoleic acid. Right, so that's this one right here. Now, when we're going to make cholesterol, we are going to need NADPH. This always occurs in the cytosol, and you're going to have 4 stages. It takes a lot of energy, high energy bonds, to generate cholesterol. So in general, there are 4 steps. And so again, it all starts with acetate. Okay, so in this case, you have to synthesize first melalonate, and that is basically 3 acetates. And then you're going to convert that to 2 active isoprenes. So here's your active isoprenes that have phosphate groups attached. And then you're going to condense 6 of those isoprene units to form a squalene. And you can see the squalene, your ring structures are starting to come together. And then the squalene gets converted to the steroid nucleus. So in the first stage is going to be, mevalonate from acetyl-CoA. Okay? So this is going to, you're going to take acetyl-CoA and you're going to add another acetyl-CoA and you're going to make acetyl-CoA. This is not the rate limiting step. This acetyl-CoA can go somewhere else and do something else. Now, when you add the third one, that's when it becomes the rate limiting step. Okay, so this is by your synthase. So your hydroxy beta methyl gluteryl CoA synthase. Oh, it's right here. So HMG stands for beta hydroxy beta methyl gluteryl CoA. And so this is the rate limiting step. Okay, so once you have this beta hydroxy beta methyl gluteryl CoA, you then are going to reduce it. This requires 2 NADPH, and you're going to generate mevalonate. The HMG-CoA synthase is going to be distinct from the mitochondria synthase that was part of ketone body production. So remember, mitochondria has its own beta hydroxy beta methyl gluteryl CoA synthase. It's in the mitochondria. This is cytosolic HMG-CoA synthase. Now, the commitment step is going to be important point of regulation. And the commitment is going to be in this reduction of HMG-CoA to mevalonate. So keep in mind, there is a difference between rate limiting and commitment step. So the commitment cannot be reversed. Okay, so once you get mevalonate, you have to go on to make cholesterol. Since this is the commitment step, your HMG-CoA reductase is going to have to be controlled at many levels. So the first level of control is going to be at the level of transcription. So we have in our endoplasmic reticulum, so ER, we have sterile regulatory element binding proteins, which are these. So sterile regulatory element binding proteins have a DNA binding domain attached to them, and they are going to actively regulate the expression of more than 30 genes. Okay, and so this DNA binding element is held sequestered in the ER by the sterile regulatory element binding proteins. Now, these binding proteins are going to enhance transcription by binding to the sterile regulatory element, or SRE. Now, when the sterile regulatory element is bound, the rate of transcription is going to increase, and that's shown here. Now, how does this happen? Well, in the presence of cholesterol, you have a sterile regulatory element binding protein cleavage activating protein, which is called SCAP. This will bind to cholesterol. And when cholesterol is bound, SCAP will interact with the sterile regulatory element binding proteins. The interaction of SCAP with the sterile regulatory element binding proteins releases the DNA binding domain. And the DNA binding domain is going to go to the nucleus, and it's going to activate gene transcription. Now, another way that this reductase is going to be regulated is through actual degradation. So if you have an increase of sterols, this causes proteolysis or degradation of the actual reductase. Because when you have too many sterols, you don't need to make more nucleus, and you want to get rid of this reductase. Another is by covalent modification. So the reductase is going to be regulated by phosphorylation and dephosphorylation, surprise, surprise. So elevated glucagon levels is going to increase phosphorylation and inactivate it. You don't want to make cholesterol if you're in a fasting state. So the enzyme that phosphorylates the reductase is called an AMP-activated protein kinase. So the AMP-activated protein kinase becomes active, and then the AMP-activated protein kinase will phosphorylate your HMG-CoA reductase and cause it to be inactive. And when cholesterol synthesis decreases when ATP levels are low, because this is AMP driven. Now, alternatively, in the presence of insulin, you're going to activate a phosphatase, which will, of course, remove that phosphate and make the HMG-CoA reductase active. All right, so now that we have mevalonate, we can make our isoprenes. So this is considered stage 2. So in the second stage, you're going to get 3 phosphate groups transferred, and these are going to come from ATP to mevalonate. And these are going to activate the carbon 5 and the hydroxyl group on the carbon 3. So again, you have mevalonate, you have ATP, and you're going to phosphorylate, and then you're going to add another ATP, and you're going to get a pyrophosphate. And then on your pyrophosphate, you'll add a third ATP. And then you're going to get a 3-phospho-5-pyrophosphate mevalonate. So this is your number 5 carbon. And over here, of course, you have your 3 carbon. And this is your 5. There it goes. Now, after you have the phosphates attached to carbon 3, and you have your phosphates attached to your 5. Your 3-phospho-5-pyrophosphomalolate is going to be removed along with the carboxyl group that is attached to carbon 1. So this is going to be your leaving carbon dioxide. So that's what's going on here. And you're also going to lose that 3-carbon phosphate group for that energy required to do that. And then you have your delta 3 isopentenyl pyrophosphate. And so this is going to, the removal of that carbon dioxide is making a double bond at the delta 3. And this is the first of the 2 activated isoprenes. The second one is going to be dimethylallopyrophosphate. And so they can interchange between each other. Oops. So stage 3 is where you're going to condense 6 of these isoprenes, and you're going to form a 30-carbon squalene. So this is called head-to-tail condensation. And it's going to involve both of the 2 activated isoprenes, the isopentenyl pyrophosphate and the dimethylallopyrophosphate. 1 pyrophosphate group is displaced and a 10-carbon chain known as granule pyrophosphate gets generated. Now, the granule pyrophosphate is going to undergo another head-to-tail condensation, and this time it's going to be with the isopentenyl pyrophosphate. And this is going to result in a 15-carbon farnesyl pyrophosphate. Okay, and then 2 molecules of farnesyl pyrophosphate undergo a head-to-toe fusion. And then both in this head-to-toe fusion causes both of the pyrophosphate groups to leave. So you this time leave 2 pyrophosphates, and then you're going to form a squalene. All right, so squalene monooxygenase adds a single carbon atom to the end of squalene molecule, and this will form an epoxide. Oops, where's my guy? Sorry. Okay, so it's going to add an oxygen there. And this is going to require NADPH. And then you're going to have unsaturated carbons of squalene 2, 3-epoxide, which is what forms, aligned in a way that they're going to convert or to have a conversion of a linear squalene epoxide into a cyclic structure. And this results in lanosterol. Okay, this takes 2 steps through a cyclase. Then you have a series of complex reactions which lead to the formation ultimately of cholesterol. All right, now when you have cholesterol, there can be several fates of cholesterol. You can make hydroxysterols. You have 2 hydroxyl groups. You can make cholesterol esters, which is important through this acyl-CoA cholesterol acyltransferase, ACAT. We'll talk more about ACAT. And ACAT is going to bring in another fatty acyl-CoA, and you're going to leave with a CoA. You can make steroid hormones, or you can make biosalts. Taurochoic acid is such a bile acid. So almost all mammalian cells, as I said before, can make cholesterol. And most of the biosynthesis of cholesterol is going to, however, occur in the liver. So a bulk of cholesterol is going to be secreted by the liver as esterol salts or in bile or as bile acids. So you have in the liver a majority and then bile salts are going to be stored in bile. Oops, it's spelled. Now, cholesterol ester production in the liver is going to be by that ACAT, and this is also ACAT is going to be important in our lipoprotein particles to make sure that the cholesterols that are taken up are esterified, because most cholesterol in our blood is esterified. So cholesterol esters are going to be packaged into the core of the very low density lipoproteins. They are hydrophobic. The tissues will pick up the esters and the tissues can use them. They can use them for their membranes, to make steroid hormones, if that is their job. It also can be used for the synthesis of vitamin D. And then esters also can be stored in the liver. Let me put 3 here. Now, your liver cells are going to make cholesterol, and the cholesterol is also going to be important for the synthesis of bile salts and bile acids. Now, keep in mind, cholesterol is going to be recycled. So, do I have this? Yeah, okay. So, free cholesterol move this over here. So you have free cholesterol that enters the gut via the biliary tract. And then this is going to mix with your dietary pool of cholesterols. And then 55% of this free cholesterol that was released is going to be reabsorbed. On a low cholesterol diet, your liver will make cholesterol. Okay, about 800 milligrams needs to be made a day. On a greater increased cholesterol intake, your cholesterol will suppress the production by those mechanisms we talked about. It's not going to allow for the generation of more enzymes. It's going to cause your reductase to be degraded, etc. So this is called feedback repression. Now let's just talk a little bit about the synthesis of cholesterol to our bile salts. So we're going to have cholesterol conversion to cholic acid and chenocholic acid. and they're going to be synthesized by the reactions that are going to hydroxylate the steroid nucleus. And then you're going to first have an alpha hydroxyl group added to the 7-carbon. So that's shown here. And you have a 7-alpha hydroxylase. And the 7-alpha hydroxylase is going to pick up electrons, and this is also part of a P450 enzyme. If you have biosalts, it's a feedback regulation, so the biosalts will come back and negatively regulate the 7-alpha hydroxylase. Now, in other steps, you're going to have a double bond reduce, hydroxylation may occur, conversions, etc. So many different steps can occur. And then you have 2 sets of compounds produced. You have a cholic acid and chenodeoxycholic acid. And the pK of the bile acids is approximately 6. In the intestinal lumen, the pH is about 6. So in the intestinal lumen, about 50% of your biosalts are ionized. So oftentimes the terms biosalts and bioacids are interchangeably used, but a biosalt actually refers to the ionized form of the molecule. Okay, so let's talk a little bit about the conjugation of biosalts. So you have your cholic acid, pKa of 6, and then you're going to have a hydroxyl group at the end of the side chain activated by coenzyme A. And coenzyme A can react with either glycine or taurine, and this is going to form the known conjugated biosalts. So coenzyme A can either work with the taurine or a glycine. And then you're going to form the 2 biosalts, taurocolic acid and glycocholic acid. So glycocholic acid and glycochenocholic acid, the biosalts are going to be conjugated with glycine. And then taurine conjugates, taurocolic and taurochenocolic acid, you're going to conjugate with taurine. Okay, so with respect to our glycocholic, and our glycocheno, you have a higher percentage present in the ionized form in your intestine at the pH of 6. In your taurine conjugates, you have a greater percentage are ionized in the gut. I'll put 3 arrows. Okay, so the liver is going to synthesize biosalts. Your biosalts are going to be released by the liver, and it's going to be stored in the gallbladder. And so your gallbladder is going to release your biosalts when there is food. And it's going to, of course, bile salts are going to help to emulsify fats. Now, your bile salts will get reabsorbed, as I talked about. They get recycled, reabsorbed, and returned to the liver. And this is pretty efficient, about 95% efficiency. And then the rest will be excreted. So you have a pool of bile salts, and in your gut, your bacteria are able to deconjugate and dehydroxylate bile salts. So you have your primary bile salts, which are cholic acid, chenodeoxycholic acid, and then you have your secondary bile salts, deoxycholic acid and lithocholic acid. Okay, so let's talk just a little bit about transport. This is another important thing about your cholesterol. So cholesterol is insoluble, and it must be transported in lipoproteins. Now you have in your lipoproteins, you're going to have your phospholipids, you're going to have cholesterol, you're going to have your tags. And so your charges of nitrogen of your phospholipids, you're going to have phosphatidylcholine, phosphatidylethylonine, or phosphatidylserine, are going to form an ionic bond with the negatively charged hydroxyl ion of the environment. So your phospholipids are going to interact with your aqueous environment. And then your free cholesterol molecules are going to be dispersed throughout the lipoprotein shell. So major carriers of lipids, of course, are going to be your chylomicrons from fed state, very low density lipoproteins made by your liver, and your high density lipoproteins, which we haven't talked about too much. So your transport of cholesterol by these different blood lipoproteins is going to depend on what the state is of your individual, fed state, fasting state, etc. And so you have in your gut, you are going to form your chylomicrons, and the chylomicrons are going to enter and release free fatty acids, and this can go to adipose tissue, etc. You're then going to have a chylomicron remnant, which will be taken up by your liver, and those pieces will be degraded. You're in a fed state, so your liver, in addition, is going to make very low density lipoprotein particles that get released, and the very low density lipoprotein particles will drop off their fats at different tissues. And these very low density lipoprotein particles will become intermediate density lipoproteins, which will release more free fatty acids, and then they will become low density lipoproteins. Low density lipoproteins will circulate, and they can become what are called bad fat. Your macrophages can eat these low density lipoproteins and form foam cells, which can start a plaque. Low density lipoproteins will also circulate in your blood and be picked up by the low density lipoprotein receptor on the surface of your liver, and the liver will take it up. Your high density lipoproteins are made also by the liver. They can also be made in the gut. They have a role in what's called reverse cholesterol transport, which we'll talk about. So you have enzymes that are going to be required for your lipoprotein metabolism. And they also act as ligands that target that are going to be targeted to specific receptors in peripheral tissues. A lot of them, of course, are going to go to the liver. 10 principal apoproteins have been characterized, and the lipoproteins are distributed among 8 different categories. So these apoproteins, we have, of course, your very low density lipoproteins that have ApoB100. You have your, I'm trying to find your chylomicron here. Oh, here's your chylomicron. Here's your chylomicron. Your chylomicron is going to have ApoB48. It's not being shown here. That's what they have. You have ApoE, ApoC, and these are all going to be important for either function or targeting to other cells. For your high-density lipoproteins, you have a nascent high-density lipoproteins, which is going to have ApoA2. It's also going to have ApoE, because remember, it's going to transfer that to your very low density and your chylomicrons. And then you have a mature high-density lipoprotein particle as well. So just really fast about the chylomicrons, we talked about these already. They are the largest. So just a few things. Largest lipoprotein. Least dense, because they have tags. They're rich in tags. Not so much in other components like cholesterols. They are synthesized from dietary lipids, so they're going to be synthesized. And they're going to enter bloodstream through the left subclavian vein. Okay, and they are going to express ApoB48, ApoC2, and ApoE. ApoC2, of course, activates lipoprotein lipase, and ApoE and ApoB48 are going to have liver receptors on the liver. And so those will help to get chylomicrons out of the circulation. All right, very low density lipoproteins, these are going to be made by liver when you have excess of sugars, fats, excess of carbons. And it's usually in the fed state. You're going to have, in your very low density lipoprotein particles, you're going to not only have your fats, you're also going to have cholesterol esters, you're going to have different apolipoproteins, phospholipids, B100s on the surface. You're going to have ApoC2 as well. You're going to have ApoE. These, of course, are given to them by HDL particles. And then these are going to, of course, go from the liver, go to the bloodstream, drop off their package to mostly adipose tissue. And it has the same fate as the chylomicron. The very low density lipoprotein remnants, which are the IDL and the LDL. 50% are going to be removed via ApoE to the liver. Okay, so let's just talk a little bit about intermediate dense lipoproteins, IDLs, and our LDLs. About 50% of these remnants of our very low density lipoproteins are not removed from liver, or not removed from the blood by liver. Instead, they have an additional core of tags and these become your IDLs. And then your IDLs will have some removal. Oh, sorry. And this will be your hepatic triglyceride. Lipase. And you'll have some fatty acids removed. And then this is going to lead to an LDL particle. And LDLs are rich in cholesterol. About 60% of your LDLs will go back to the liver. And they bind to a LDL receptor. And then they're going to be removed from the circulation. Cholesterol inside of this is going to be converted to biosalts or incur in the membrane of the liver or be reesterified. And that is again by that ACAT protein, that transferase, and this can be stored or released. Packaged in another very low density lipoprotein, for example. The remaining 40% of these are carried to what are referred to as extrahepatic tissues, and these are tissues that need cholesterol. These are going to have ApoB100 receptors, and then they're going to use the cholesterol to synthesize steroid hormones. Okay? So this is just another overview picture. Seen this many times before. Some things to point out is that, again, you have your, in this case, we have a very low density lipoprotein particles. Lipoprotein lipase through that C2 releases fatty acid and glycerol. You have muscles and adipose tissue that pick up the fats. Muscle's going to use for energy. Adipose tissue is going to store the energy. You then have your very low density lipoprotein particles will become intermediate density lipoprotein particles. These can go back to the liver, where the liver has receptors for these, and they can bind to those, and then you can make them into different things in the liver. Your intermediate density lipoprotein particles will have high in tags, and then they'll use that hepatic triglyceride lipase to remove those tags, and then it becomes a low-density lipoprotein particle. About 60% of your low-density lipoprotein particle will go back to the liver, and about 40% will go and donate the cholesterol to tissues that need cholesterol. And those other tissues are going to pick those up. Your low-density lipoprotein particles can also be oxidized and be picked up by macrophages to form foam cells, and these can lead to lesions in the blood vessel. Okay, so we also have our high-density lipoproteins. So our synthesis of high-density lipoproteins is going to be done in 3 ways. The first is going to be just simply a liver cell making a nascent high-density lipoprotein particle. And this is a very small amount. Okay. Your high-density lipoprotein is relatively small, and its shell is going to contain phospholipids, free cholesterol, and a variety of apoproteins. So on its surface, it's going to have ApoE, it's going to have ApoA1, it's going to have ApoA2, it's going to have ApoC1 and C2. So we'll just add our 1 and 2. 1 and 2. A second method of making a high-density lipoprotein particle is a buddying. It's what's a buddying of apoproteins from chylomicron and very low-density lipoprotein particles as they're digested by that lipoprotein lipase. And the third method is going to be when there's free apoprotein A1, and A1 will acquire cholesterol and phospholipids from other lipoproteins. So it kind of builds itself. Okay, so it's making a new HDL particle. Then you have to have maturation of these nascent HDL particles. So they are going to accumulate, accumulate their phospholipids and cholesterol. And this is typically from cells lining blood vessels. So they just pick things up. As in, it's going to have a central hollow core that will accumulate cholesterol. And they're typically in the form of esters. HDL particles have to contain ApoA1. And they will also have another enzyme, which is a lecithin cholesterol acyltransferase. So LCAT is going to be important for the formation of cholesterol esters. Oops. And this is going to be from lecithin, which is simply phosphatidylcholine, with cholesterol. So LCAT is going to be on the surface of your nascent HDLs, and it's going to interact with cholesterol and phosphatidylcholine, which is typical cell membrane phosphoprotein or fatty acid. And then the phosphatidylcholine could come from the membrane of chylomicrons, or it could come from very low density particles, etc. And then the LCAT is going to make cholesterol esters that are going to form the core of the high density lipoprotein particle. Now, the third thing that has to happen is high-density lipoproteins are going to help a cell that is overburdened with cholesterol. So they have the ability to remove cholesterol. And then the cholesterol goes to the liver. And this is reverse cholesterol transport. Right? Now, how does this happen? Well, this is important because remember, cholesterol is a big part of the cell membrane. But if cells pick up too much cholesterol, they have nowhere to do it. You can't break down cholesterol, so they just stick it in their membrane. And so they become cholesterol laden. And so in vascular tissue in particular, you don't want a buildup of cholesterol membranes because then it can get released, picked up by foam cells, and can start cardiovascular diseases. So removal of cholesterol from the cell, and then you want to transfer that to the lipoprotein. In this case, it's going to be high-density lipoproteins. So this is your removing and you're transferring it to high-density lipoproteins. Now, the cells themselves are going to contain a particular protein, right here, ABC1. Let's see. So ABC1 just simply means ATP for the A part, binding for the B part, cassette for the C part, protein 1, ABC1. And so ABC1 is going to use ATP to move So it's simply moving cholesterol from the inner leaflet. So here we have our cholesterol. See, it's on the inner leaflet. And you have a free cholesterol. It moves it from the inner leaflet to the outer leaflet. And then you can simply have your APO1, A1 protein, which is going to interact with them and generate this nascent HDL particle. Once the cholesterol has reached the outer membrane, the HDL particle can then pick it up and accept it. It cannot, of course, take a cholesterol that's on the inner leaflet. So the ABC1 is important for moving cholesterol from the inner to the outer leaflet. Now, to trap the cholesterol within the HDL particle, the HDL particle has the LCAT enzyme that it picks up from circulation to form a cholesterol ester. So you basically have a cholesterol, and then through lecithin, phosphatidylcholine, you generate a cholesterol ester. And cholesterol esters migrate to the core of the HDL. So if you have elevated levels of lipoprotein-associated cholesterol in the blood, particularly LDLs, they are associated with the formation of cholesterol-rich plaques in the vessel walls. High levels of HDL in the blood are believed to be vascular protective because increase of the rate of reverse cholesterol transport away from the blood vessels. So increase of LDL, which is called bad cholesterol, can lead to the formation of plaques. Increase of HDL, or good, takes cholesterol away from blood vessels, and it goes to the liver, and then the liver can deal with it. So what's the fate of the cholesterol? The primary clearance of HDL from the blood is through its uptake by scavenger receptors. And these are scavenger receptor B1, which is on many cells. When a HDL particle is depleted of cholesterol and esters, it dissociates. Cholesterol plus cholesterol esters. It dissociates from the receptor and goes back into the circulation and picks up more cholesterol. HDL is going to interact with other particles. We already know that it's going to interact with your very low density particles and chylomicrons. And it gives them the ApoE and ApoC2, we know that. When the high-density lipoprotein gets free cholesterol from cell membranes, these get esterified, and that's via that L-cat reaction. All right, so reverse cholesterol transport is going to be when your high-density lipoprotein particles are going to transport free cholesterol to the liver, or going to transport your cholesterol to a circulating very low-density lipoprotein particle or a very low-density lipoprotein remnant. And this is mediated all by cholesterol ester transfer protein. So the HDL will transfer and this is in exchange for tags. So the greater the concentration of your rich lipoproteins in the blood of tags, the greater this exchange. So the more tags, the more exchange. Mature HDLs are equal HDL3 after this CETP reaction. So they lose cholesterol, gain tags, and then when they lose cholesterol and gain tags, they are HDL2. Okay, so why would you want to do this? Why would the high-density lipoprotein want to switch over and pick up tags and give up cholesterol? So you can think of this because the high-density lipoprotein scavenger receptors are in many cells. So if the high-density lipoprotein simply just transfers cholesterol back to cells, they become cholesterol laden again. The very low-density lipoprotein and its remnant have receptors on the liver. The very low-density lipoproteins can transfer those cholesterol esters back to the liver, and the liver can deal with the cholesterol either by storage, making bile salts, etc. All right, so this is just showing this. So we have in our liver makes our high-density lipoprotein particles. Change that color. Our high-density lipoprotein particles is going to have ApoA, especially ApoA1 is very important. C2 similar in E. And of course, it can mature your chylomicrons by transferring those C2 and E. It can mature your nascent very low density lipoproteins by transferring, oops, sorry, not E100. It's going to transfer the E and C2. Your high-density lipoprotein particles can pick up cholesterol from cell membranes, and then those cholesterols can become esterized through your transferase, and then they make cholesterol esters. Your cholesterol esters can be transferred to very low-density lipoproteins in exchange for tags, and then your very low-density lipoproteins can eventually get the cholesterol back to the liver, and the cholesterol in the liver will be made into bile salts, etc. All right, so just a little bit on lipoprotein receptors. The best characterized lipoprotein receptor is the LDL receptor. Now, the low-density lipoprotein receptor is going to be specific for ApoB100. It has what they call a mosaic structure. It has 6 different regions. The first region is going to bind to ionic calcium. And then when the side chains are protonated, the calcium is released and it leads to a conformational change that allows the LDL to disconnect from its receptor docking site. So calcium is important for before you have receptor engagement. The lipoproteins are going to bind to the receptors, and the receptors are going to be in your clathrin-coated pits. And these clathrin-coated pits are going to enable the uptake of the receptors by endocytosis. The apoproteins are going to have a ligand for receptors on specific target tissues. Typically, the most common are going to be, of course, liver, the adrenal cortex, as well as gonads. Lysosomes in the cells, the cholesterol esters of the low-density lipoproteins will be hydrolyzed, and they will form free cholesterol. Your lysosomes don't break down cholesterol like they would break down lipids and proteins. The newly esterified cholesterol contains primarily oleate or palmitoyl oleate, and these are different than the cholesterol esters in LDLs. LDLs usually have linoleate, as I talked about before, or polyunsaturated fatty acids. So synthesis of LDL receptor is feedback inhibition. And this is through the increasing levels of cholesterol within a cell. There's also an LDL receptor-related protein, and these are going to be related but are going to bind to a broad spectrum of ligands. So they're not as specific as the B100. So it's going to bind to lipoproteins. It's going to bind to alpha-2 macroglobulins. These inhibit blood proteases. And tissue plasmalogen activator. It will also recognize ApoE. This is going to be found abundant in the cell membranes of liver, brain, and placenta. We also have a macrophage scavenging receptor. These are going to be particularly high in our phagocytic macrophages. That's my symbol for macrophages. They are non-specific receptors, so that's why they're called scavenger receptors, and they're going to bind to different types of molecules, including your LDL particles. LDLs first have to be modified in order to be bound by the macrophage scavenging receptor. So modification of LDL is natural. It occurs naturally through the metabolism of these lipid particles, and it's going to involve oxidative damage. They are lipids, so they get oxidatively damaged. And then the macrophages can bind to these oxidatively modified LDL particles. And then when the macrophage becomes engorged, they become foam cells. Accumulation of foam cells, especially in subendothelial space of blood vessels, is the earliest evidence of developing an atherosclerotic plaque. This is called a fatty streak. So oxidation of their lipoproteins is going to be the same thing that was happening in our cells. It's going to be superoxide radicals, nitric oxide, hydrogen peroxide, etc. And remember, those are inside of your cells. They can also be outside of your cells. And so they're going to do damage to these particles in the phagocytic cells via their scavenger receptors, can pick up these damaged particles and try to digest them. So the aspects of atherosclerosis. So you have a normal artery, so you have three distinct layers, as you already know: the intima, subintimal extracellular matrix, and this is where you have the smooth muscles. And then you have the tunica media, which is the middle layer, the lumen. And so the initial step in the development of a lesion within the wall of the artery is the formation of a fatty streak. And remember that the fatty streak is just simply an aggregate of foam cells, of your macrophages. Okay, so and this basically means you have an accumulation of lipid-laden macrophages in the subintimal space. And this looks like a yellow-white linear streak. Now, eventually you'll have your plaque form, and it's going to form with the small thrombus, and this is called a fissure. And then you can have a healed fissure, or you can have a non-occlusive fissure form, a thrombus. This can then be healed again, and the thrombus is going to be suppressed. Or you can have an occlusive thrombus form. Now, risk factors are going to be elevated blood pressure, arterial blood pressure, hypertension, circulating levels of particles, specifically your LDLs, even your chylomicron remnants, VLDLs, or low levels of HDLs are going to be risk factors. Cigarette smoking, chronic elevations of blood glucose levels, because that's going to lead to dysbiosis of your metabolism. High circulating levels of angiotensin II, which tend to cause cells to secrete adhesion molecules, and adhesion molecules will allow your monocytes, which are circulating macrophages, and slow them down so they will bind to the blood vessels. Okay, and so regulation of cholesterol. So let's go back a little bit to cholesterol. So cholesterol, remember, is going to start with acetate. And then you're going to go through several steps and eventually get to HMG reductase. And this, of course, is going to be irreversible and lead to mevalonate. So this is going to be positively regulated by insulin, and it's going to be negatively regulated by glucagon, negatively regulated by AMP kinase, and negatively regulated by oxysterol. Okay, so where does oxysterol come in? Well, mevalonate, of course, goes on many steps to make cholesterol. Right. And then you have a transferase to make your cholesterol esters. And then cholesterol will be a positive regulator of this transferase. Now, alternatively, if this starts to build up, you're going to lead to oxysterol, which is going to increase. And then oxysterol is a negative regulator of making more because it means you have too much cholesterol and it's building up. All right, so this is a summary of this. There's a lot of information in here. But in general, remember, cholesterol is going to be made from acetate, acetyl-CoA. All of the carbons are coming in that way. You're going to have to make some intermediates. There's going to be several steps. You have your isoprene units that are going to eventually form squalene. Cholesterol is going to be made, converted to an ester. That's going to be packaged in your lipoprotein particles. You have different lipoprotein particles, chylomicrons for diet, VLDLs for excess carbons, energy. HDLs are going to be made to mature your VLDLs and nascent chylomicrons. You're going to have your VLDLs break down to IDLs and LDLs. LDLs are going to be important because those are the ones that are going to take cholesterol to organs that need it to make your steroid hormones. You also have a reverse cholesterol transport. Your HDLs can pick up cholesterol from cells. You also have your regulation of cholesterol, especially of the reductase. That could be at many levels. So there's a lot of information in here.